This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1259, recorded on October 3rd, 2025. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Western Massachusetts, Alan Dove.
Good to be here. Here in Western Mass, it's 72 Fahrenheit right now. Pretty clear skies, and that's 22°C. Very, very nice weather. Just a little bit of fall in the air, and then it's supposed to warm up again and then cool off again.
It's currently 23°C in Chelsea, but this morning on the train platform, it was 8°C. Oh my gosh, 8 degrees above freezing. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. It is 74 degrees Fahrenheit in Madison, and similar to the way Alan described, very nice. I took a walk at lunchtime, and it was a lovely walk. It's a nice fall.
Walking is good. I appreciate walking. On my travels, I walked a lot. But my hotel in Greece was a 1.5 kilometers from the conference center. I used to walk back and forth. Nice. It was really good. It was sunny and warm. With the ocean right there. It's lovely. If you enjoy these programs about science, real science, truthful science, hypothesis-driven science, fact-driven science, not science because I feel it should be that way, as in, I feel we should split the vaccine doses up only because I feel that way. I feel it. Oh my God. We don't do that here. We have feelings about other things like food or the weather, but science is driven by fact and experiments and observations. And if you like that, we'd love your support to be able to continue them. We do need to tell people about science in these times when confidence in science is not great, especially in public health and vaccines. So go to microbe.tv/contribute. Number of ways you can support us. We'd really appreciate it. Couple of news items here, not your usual news, but articles related to what's going on. I found this one in Nature. It's by Melanie Ott, Olivier Schwartz, and Alex Siegel. Public trust in science has declined since COVID. Virologists need to unite around safety standards. Now, this is an opinion piece. It's a comment, right? So it's the author's opinions. So I wanted to express my opinion about it.
Yes. I will say, before you express your opinion, that when I saw the title, I was expecting one type of article. And really? Then when I read the article, it was not at all what I would have expected. So I was expecting sort of data about public trust in science and data about changes in public trust in science. And this seems to be an opinion about virologists need to unite around safety standards, sort of the second part of this. And it's really about, hey, virologists, get your act together and think about safety standards.
Yeah. So here's my feelings. So the first part, they're talking about how I think they're wrong on why people have lost trust in science. It's not because the Wuhan Institute of Virology worked at BSL-2 conditions. It's not because virologists don't have safety in mind. It's because a few very loud people have made everyone think that virologists are cowboys and we're not safety-minded. People like Mark Lipsitch and David Relman and Richard Ebright, they don't understand that we are safety-minded, and they don't understand the experiments that are trying to be done, and they lie to the public, basically, especially Ebright and now Bhattacharya and Alina Chan.
Well, and this is exacerbated by the current political environment where this kind of thing plays very well and is used to reinforce the identity of an in-group that will vote for these clowns.
I kind of think that's why I didn't link the two together when I first read the article, is that, yes, I agree that there are some issues with public trust in science, but if I were to think about the reasons behind it, I'm not sure how many members of the public know this level of detail about safety standards that is discussed in this article.
This may be on the list, but it's way, way down it.
I mean, the idea that you should have an international safety standard for different biosafety levels for different pathogens is just not going to work, because every country has a different culture. They have different immunity. They have different pathogens, and you cannot expect everyone to have the same approach. So
I agree with where you're going with that it would be a very hard thing to arrange and coordinate, but there are international regulations for a lot of things that are harmonized between countries. And the way it's done is every country makes their own regulations and then, consults with the other countries about how their regulations are, and everybody tries to make them about the same. So I don't think it's impossible to have something like an international set of standards. And we kind of have that with the BSL 1, 2, 3, 4. They're mostly the same most places. But I see what you're saying, that's not going to suddenly, change trust in science.
No, I think nobody's going to pay any attention to that. When it gets done, there won't even be a news article about it because it's not negative, right?
Right.
Well, and because it's too involved.
Yeah, it'll be far back in some appendix of the newspaper.
Yeah. And another thing they suggest is that, so this idea that virologists are not safe is just offensive, which is promulgated by the Ebrights and the Relmans and the Lipsiches. They say we work without safety and we're not allowed to police ourselves, but they're not virologists. They don't know about how the systems work. There's a lot of safety involved. Let's say we do BSL 2, 3, and 4, and we're very conscious about being safety. And there haven't been any accidents, at least in the U.S. So, and don't start telling me there are accidents. When Ron Fouché told me once, when their computer crashes, they have to write it down as an accident. Okay, so they have to monitor everything. If the temperature is off, they have to. So there have been no accidents that have released pathogens because it's a good culture of safety. So I find it, and as we have already said, that this is not the reason why public interest in confidence in science has declined.
Also, who would work there in an industry that didn't have safety? Because the virologists themselves would be the first ones at risk.
Yes,
of course. So they also advocate using AI as much as possible to predict, for example, what cells in animals could be susceptible to a virus, to which I say, good luck with that. Okay, let me give you an example. Early in the pandemic, a paper was published where they looked at the spike of SARS-CoV-2 and they predicted its affinity with ACE2 of different animal species and used that to say whether those animals would be infected or not. We did that paper on TWiV. We laughed like crazy because deer are at the bottom of the susceptibility list. You can't do it. Maybe in 20 years. I mean, that's a huge project with a lot of data involved, maybe which we don't even have yet. Right now, you need to do experiments. Yeah. Just like how NIH wants to use computers to substitute for animal experiments. Well, maybe one day, but not now.
Yeah, we don't know enough to actually model it with a computer.
Yeah. And that's not to say that people shouldn't do those analyses, but the approach would be, okay, let's see if we can build an AI model that gives us results equivalent to what we have in the experimental work.
Yeah.
And then compare it to experiments where you know the results, but the computer doesn't. You've got your training data and your test data and see how it goes. Keep working at that for a while. And I think, Vincent, your estimate of 20 years is probably
and maybe in doing that will actually teach us about, some interactions or some things needed for infection that we didn't know before. It's useful to do it, but we can't give up on all the other stuff.
I'm all for exploring new ways of doing things. It's great. But don't say we are going to replace animals with computers today, right? Which is the headline at NIH. We're going to replace, instead of saying we're going to investigate the feasibility of using computers and AI to replace animal studies in a while. And then maybe it could even replace humans and you don't need to do clinical trials. Hey, why not? I'd be all for it if you could. I think it's unlikely. I think it's like a moonshot.
And there are other technologies that people are working on. We've talked about organoids. We've talked about, these other types of systems, which can be used to supplement or in some cases even replace animal studies that used to be done. And that's great. that wouldn't satisfy these folks who are saying, you shouldn't be working with actual viruses.
Yeah. I read a paper earlier this week where some scientists had actually described kind of a new cell type that was doing some immune processes, sort of in a new immune cell type. If we don't even know all the cell types yet, I'm not sure that we can take the data we have and predict things like tropism or things that don't need us to do actual experiments.
Brianne, what constitutes a new cell type? A slightly different level of a certain cell marker on this, right? I mean, that's the problem. You guys have, it's twofold higher in this and threefold lower. It's a new cell type. I don't know where you draw the line, right?
That's the T cell problem, but this wasn't a T cell.
So that's just for T cells. So B cells are all pretty homogeneous?
I have an answer to that, but my B cell biologist friends would yell at me if I said it.
That's okay. I understand that it's complicated. But yes, if we don't know everything, it's hard to... So a computer can only do what you give it, right? It's not going to be creative. Anyway, I don't agree with the article. It's fine that you put your opinion out there, but for those reasons, I'm not really enthusiastic about what it says. Okay.
I do like that they led with a stock image of a plaque assay.
Yeah.
True.
It's good. Okay, now Alan have you put this one in here?
I did
Alan?
put this one in here. Meanwhile, in Canada, so the Mounties showed up at an ostrich farm. No, this is not a joke. And confiscated all the ostriches because there's this case going on. This ostrich farm got infected with H5N1 avian flu. Bunch of the birds died. And the Canadian regulations say that if you have poultry, and these ostriches are classified as poultry, and you have an outbreak of avian flu, you kill the birds, right? Fairly straightforward. This has been going on chicken farms in the US and Canada and elsewhere. The owners of this ostrich farm, I guess, were able to pull some strings and get more people and public relations campaign because ostriches are cute. And now there's this whole brouhaha, and the thing has gone to Canada's Supreme Court, which will decide whether they can get out of culling these ostriches. The farm owners are arguing, well, the surviving ostriches have now survived this. They've got immunity to it. They're not going to be infected. They're not posing a risk, so we shouldn't have to kill them. And they're making all sorts of arguments about the potential of these birds for research, which I'm not even sure how far along any of that is. Of course, the usual circus clowns have logged in. RFK Jr., Dr. Oz. I think Mehmet Oz actually offered to take these ostriches and put them on his ranch in Florida, because of course he's a Florida man.
Yes.
So all this is going on, and the beleaguered Canadian Food Inspection Agency now has custody of these ostriches, but they cannot get rid of them until the Canadian Supreme Court rules on this.
Why don't they just test them for H5N1?
I don't know why that's not an answer at least it's not been discussed in the coverage I've seen so far. But yeah.
Because I think it's unlikely that they're positive so long after the initial infection, but you should just test them. It's not that expensive. I thought initially the farmers would not let them come and test because if any of them were positive, they'd have to cull the whole thing, right?
Yeah.
And that would just reinforce the ruling. But
yeah.
It seems now that they've taken the birds, they could just do a little swab and test it. It's not hard, right?
Yeah.
I don't get it.
But I don't know if that would address the issues in this case. So
yeah.
Well, the idea is that as you say, if a herd is infected, you have to cull it, and you can't you can't go around that. It's not right to go around a regulation.
Exactly. If the rule is you have to cull them, then you have to cull them.
Yeah, there shouldn't be special exemptions just because you managed to get a lot of hits on TikTok. Yeah.
Exactly right. So we'll see. It's a very interesting story, though,
yes.
Right? How it's developed. I mean, I like the ostriches. I like the fact that they make big eggs full of antibodies that people think might be useful. Okay, but they're not the only ostriches in the world, right?
Yes, actually, I enjoyed this sentence in the article that was about that. They say, with some contending that copious antibodies found in the bird's enormous eggs could provide a font of new products to improve human health and beauty.
That was a beauty part. What does that have to do with anything?
Right. No, I guess maybe that's their lead product or something.
Yeah, maybe you're going to make some sort of face cream.
Yeah.
From...
Oh my gosh. But, I mean, you can make nanobodies in alpacas, right? And,
yeah.
You could. I mean, now you don't need ostriches.
And you can get similar antibodies from chicken eggs, and there are, as you pointed out, many, many, many other ostrich farms in the world. This is not some unique population. They're not genetically engineered ostriches that are going to be irreplaceable.
Yes. Just test them, they'll be negative, and they can go home. Okay?
Maybe.
Yeah.
Or if the rule is that they were supposed to have been culled, then either change the rule or cull them.
Yeah.
Although, I have to say that, for RNA viruses, that pieces of RNA which are not infectious can stick around for a long time, right? Not necessarily infectious. So what are you going to do? If it's PCR positive, it doesn't mean anything really.
Right.
So far after the acute infection is over. So in which case you would have to look for infectious virus, which is not hard to do for influenza viruses, right? But all right, that's our 2 cents.
Yep.
3 cents. Now let's do some science.
How many cents is it Canadian?
That's a good question.
Yes.
Actually, that's all science. This is continuing with the literature now. We have an article in Cell Reports, which is entitled Dairy Cow and Avian Origin Clade 2.3.4.4b H5N1 Induce Severe Mastitis in Lactating Goats and Transmission to Suckling Goats.
And speaking of Canada, so the first author is Tamiro Alki, senior author is Johannes Berhain. These folks are from the Canadian Food Inspection Agency in Winnipeg. I guess there with the ostriches, maybe. University of Guelph, Ontario, Defence Research and Development Canada in Ottawa, Public Health Agency of Canada in Winnipeg, Sunnybrook Research Institute in Toronto, University of Toronto, St. Jude's Children's Research Hospital in Memphis. I guess that's Richard Webby, who's been on TWiV. Mm-hmm. University of Saskatchewan in Saskatoon, and University of Manitoba, Winnipeg. So a mostly Canadian cast here.
So as everyone should know, if you're listening to TWiV, the H5N1 avian influenza viruses for many years have been a little scary to people. They cause mass mortality in various wild birds and domesticated birds. And not too long ago, a new clade emerged from a reassortment event. It's 2.3.4.4b. And these viruses are very unusual, H5N1, because they infect many new kinds of birds and mammals. And they persist year-round. They're not just seasonal. So they've moved entirely around the world. And, of course, as you know, in the U.S., they have infected dairy cows. That's the right word, right? A dairy cow?
Yes.
The cow is a female.
Yes.
And a steer is the male, is that
yes,
right?
A steer is a neutered male.
What is a non-neutered male?
A bull.
A bull. And what's the generic name for these animals?
A cow.
A cow, is it? It's not, a cow doesn't mean female?
I think cow means female usually.
Cattle is the whole thing.
Cattle
right?
is the plural.
So cattle is male and females together.
I feel like that's
maybe
a general.
Cow is the.
Yeah, I think cow is general as well.
Okay.
Anyway,
if they're steer that are being raised for beef, they're beeves.
Mm.
Okay. Anyway, this clade got into U.S. dairy cattle, and we've talked about it a number of times, and has, it affects the mammary glands. It's got into the milk, and we've had a lot of discussions about that, including with Richard Webby. But not too long ago, this virus was detected in neonatal goats in Minnesota.
Kids.
Right? Kids. And in fact, recently also a sheep in the United Kingdom. So these are additional mammalian hosts that this virus is getting into.
Now, this virus hasn't been found in dairy cows elsewhere in the world. Is that because people are not looking very hard, or do we actually believe that it hasn't crossed any borders?
I noticed that exact same thing when I was reading this paper and wondered about it as well.
I don't know the answer to that. In the U.S., it seems to travel with cow movement, right? And so it transmits from cow to cow for sure. And so I don't know if we send cows overseas. Do you know? Does anyone know?
I don't think we do. And I do think that it was sort of one spillover into the cows.
Okay, but we do send...
It was, yes.
Yeah, it was one spillover, and it's a unique virus.
I'm pretty sure that at least at some point in the past couple of years, we have had cattle exchanged over the North American borders.
Yes, that's right. That's right.
Because with screwworm now migrating up through South America into Mexico, there's been a restriction on transporting cattle across the U.S.-Mexican border. That's not to say it doesn't happen illegally, but it had to have been happening in order to have been stopped, right? So it's a little puzzling / suspicious to me when something like that happens, when a virus obeys national borders. And there is a strong motivation not to look for this.
Yeah,
right.
Of course.
Yeah, even if we say it was a unique virus, they talk about spillback into birds and other things, and the birds are not going to respect national borders, and so I can't see how the birds wouldn't carry it, if nothing else.
Well, they say here that in Pakistan they've found the virus in small ruminant livestock near poultry farms. So I think it's a matter of time before you pick it up in dairy cows elsewhere, right?
Yeah.
So anyway, they say here the cow movement is a primary driver of epidemics, and cows can serve as a reservoir, right? And so you could get virus from the cows back into wild birds, into poultry and other mammals, including humans, right? So there has been accidental exposure among agricultural workers, and they have gotten infected as well. They've gotten mild to moderate conjunctivitis and influenza-like illness. No fatalities yet reported. So these cows get mastitis, get reduction in milk production, and they say alterations in milk texture and consistency. I have to say, I'm not a big fan of milk. I don't drink a glass of milk. A lot of people do. I put it in cereal, and that's because the cereal kind of masks it. Otherwise, I don't really like the taste of milk and the consistency. But this idea, alterations in milk texture and consistency, it's kind of weird.
We'll be talking about that a little
gross.
bit in the data as well,
Yeah,
and it does get gross.
So as of June, 1,073 dairy herds in 17 states of the U.S. Were affected. And as I said, you get a reduction in milk production, but you can also get other symptoms in the cows: pyrexia, respiratory distress, diarrhea, weight loss, reduced rumen motility, and rumination. I don't know. What is rumination?
That's where they chew their cud.
Oh.
So they have 4 stomachs, and they barf the food back up into their mouth and chew it again and put it back in the next stomach.
So that's not like when we ruminate.
No, it's not when we ruminate. It's not when we chew over an idea. They're actually chewing over a meal.
That's good. So you can find milk in these cows. You can see infectious virus and RT-PCR positivity. And in fact, giving that infected milk to cats can kill the cats. I bet you don't like that, Brienne.
I do not like that.
I do not like that either. And you shouldn't be giving milk to cats anyway, because it often gives them diarrhea when they're adult cats.
Really?
Yeah.
Yes. That's why you have to buy the special cat milk.
Well, yeah, at least it should be pasteurized, right?
Also, yes.
Certainly it should be pasteurized, but my wife actually discovered that half and half in small quantities is somewhat tolerated.
That's interesting. Because it's a bucolic image of a cat
lapping at the milk.
Lapping
yeah, you can do that with kittens. Still not recommended, but you can.
Interesting.
They can, because
anyway,
adult lactose tolerance is not a common thing in mammals.
We don't actually know the extent of cow infection in the U.S. We're not sure. We're not really sure how farms initially get infected, and we don't know how goats would get infected as well. Now, milking equipment can get contaminated, right? And that can transmit. Milking operations can facilitate transmission. And they've done some experimental infections in cows. and they've shown that you can get, if you put the virus in the udder, that you get necrotizing mastitis, which you see in naturally infected cows. It's just utter destruction.
Yes.
Had to say that. Okay, so
that was cheesy.
I know, it's very cheesy. These viruses, this is very important. Now, these circulating 2.3.4.4b viruses, they bind to avian alpha-2,3 sialic acids by the hemagglutinin, okay? And some of these have a single amino acid change in the hemagglutinin that allows binding to human sialic acids, which would be alpha-2,6. And in the mammary gland of the cow, there's a lot of alpha-2,3 sialic acid, and that leads to efficient replication. And we'll be talking about that in a moment. But even though that's the case, there are both avian and human receptors in the mammary glands. So there's some concern that, the virus could be selected to bind those human sialic acids better in the future. I mean, the viruses bind alpha-2,6 kind of poorly, but that could improve by a single amino acid. So, all right. So now in the current study, they're infecting goats with two different genotypes within that same clade, 2.3.4.4b.
Yeah, so the focus of this study is how well does this virus survive in goats and what are the effects?
Yeah. Does it transmit? How does it replicate? Where does it replicate? What kind of disease and so forth? And so they have two genotypes. They have cow H5N1, which is the virus that's infecting cows. And they have a TWiV H5N1, which is a turkey vulture. So those birds that show up in your backyard and they look huge. Do you ever have any of those, Alan?
I haven't had turkey vultures here. I did see, I don't think it was a turkey vulture. It was a different vulture down in Mississippi.
Maybe it's just a turkey.
Well, we get turkeys.
So you can tell the difference between those.
Yeah, I can identify a turkey.
So they have a cow and a turkey vulture, H5N1. They're going to use both here. And the reason they do the turkey vulture, because it's quite pathogenic and can transmit by direct contact. As opposed to aerosol. In ferrets, it doesn't transmit through respiratory droplets very well. So what they do is they have two groups of 5 lactating goats per group, and they get inoculated by 2 roots with either virus strain. All right, and then after 24 hours, the kid goats are reintroduced and allowed to suckle and so forth. So you can see if there's transmission going on.
Yeah, so they're giving these goats both in the udder and in the respiratory tract.
That's
the
right.
virus.
So the cow H5N1 causes fever in all the lactating goats, and then 2 kids develop fever. So there's some transmission to the kids. They get nasal discharge. They get diarrhea. Not everybody gets the same symptoms. Some of them get diarrhea. Some of them get abnormal feces, and many of them get dehydration, very severe dehydration. They have to be euthanized because of that. The TWiV isolate is more lethal, essentially. Higher temperatures, fever spikes, nasal discharges, diarrhea, abnormal feces, also transmitted to the kids, and a lot of dehydration, lethargy as well. So it obviously they both reproduce and cause disease and are transmitted to the kids. And both of these groups show udder swelling, right, in the present, which is indicative of mastitis, which they confirm. And they also see alterations in milk quality and quantity.
And this paper is open access. You can download it, and I will warn you that figure 1 looks a little gross toward the bottom.
Yes, where you get to see the milk.
Yes. So the milk quality, what's going on here, mastitis, as the name implies, is inflammation of the udder. And with that inflammation, you get this infiltration of leukocytes, and those are going to cause a change in the texture of the milk. They describe it as ropey at one point. So they're like, these tendrils of crud, which would be the leukocytes and other immune response stuff in there. And they can also quantify this or measure the not purely quantitative, but there's a semi-quantitative test, the California mastitis test, and that's what those sets of 4 little cups are. I had to look this up. I was looking at it. What the heck is going on there? And it's a paddle with 4 little cups attached to it, and you milk from each teat of the udder, the 4 different ones, and then you can check by adding a reagent that stains and kind of coagulates the leukocytes that are in it, if there are any. And so you see there are a couple of them that are just basically clear. There's a slight pinkishness to it. And then there are others where it's just really, really clumped up, and that indicates that particular part of the udder is in mastitis.
They say in the text that milk becomes thicker, more viscous, transitioning to a buttery or whey-like consistency with visible clots or curds. And the milk production goes down. And in some animals, they actually stop making milk. And, this is important because people in many countries drink goat's milk.
Mm-hmm.
It's a huge source of protein in a lot of places.
Yeah. They looked at viral RNA shedding in these lactating goats and their kids. And they'd take a nasal swab, an oral and nasal swab. They can find viral RNA in the nasal swabs of all the lactating goats from both viruses. And they have kind of intermittent shedding patterns, depending on the goat. Some of them start early, some of them start a bit later. They actually got an infectious virus from one of the infected lactating goats. And in kid goats, the RNA was more consistently found in oral than nasal swabs, which they say maybe means exposure through, suckling virus-containing milk. They look at the dynamics of viral shedding over time for these both viruses. They make comparable titers of virus. This is by infectivity now in the milk samples. And the peak in titers can coincide with severe clinical mastitis, and then it drops over the subsequent days. So both of these 2 viruses, the cow and the turkey vulture, make high amounts of infectious virus and shedding. So again, and this is also in milk. This is why milk is a potential source of transmission. They also look at different tissues to see which ones have viral RNA in them: mammary glands, lung lobes, supramammary lymph nodes, and they look for RNA. They can find viral RNA starting at about day 3, and in most of the samples, they can find it in bronchoalveolar lavage fluid. They can find it in lungs, supramammary lymph nodes, even in the gastrointestinal tract, reproductive tissues, the uterus of goats as well, and in the brain of animals infected with the turkey vulture virus. But very high levels of RNA in the mammary glands in particular. And this starts to decline, of course, by a certain time after infectious. Now, here's some numbers, if you're a virologist and you know virus titers. So the highest viral load in the mammary glands of lactating goats at 3 days in this one animal infected with the turkey vulture, the titers were 1.78 times 10 to the 9th TCID50s per mil. That's tissue culture infectious dose 50%. And the other was 3 times 10 to the 8th. It's a lot of virus.
That's a lot.
Infectious virus. It's good. And they can recover infectious viruses also from lung tissues of these goats. They do some histopathology to see what kind of changes are going on caused by virus infection. They see a lot of, to see a tropism for the mammary glands, which is characterized by necrotizing mastitis, so necrosis of the tissue. They can see, and they describe that very, very well here. They see a lot of viral antigen and RNA in secretory epithelial cells in the goats infected with both viruses, and a lot of antigen in the mammary gland overall. So there's a lot of evaluation by histopathology, but we're not going to go into that here. Pulmonary pathology is less severe compared to mammary pathologies. So with the cow H5N1, they see a few foci of necrotizing pneumonia. characterized by alveolar macrophage infiltration and localized tissue necrosis. And interestingly, the viral RNA and antigen and RNA were mostly seen in macrophage-like cells within the lung. And so this turkey vulture virus makes a mild interstitial pneumonia, and they can find a lot of viral RNA and protein in a few bronchioles. So it's not a massive pulmonary infection.
Right.
It seems to be worse in the mammary gland.
Yeah.
How about sialic acids now? Let's take a look at that in the goat. What do you do for a living? Well, I study sialic acids in goats. And remember, caprine is another way to say goat.
It is.
Yes.
I like caprine. And I always think of capers when I say caprine.
And capricious.
Yes.
And capricious. But, capers could look like goat poop, actually.
I guess.
Oh, sure. Yeah.
Does capricious have something to do with acting like a goat? Is there
Capricious?
some... I mean, I know what capricious,
right?
I know what capricious means, but I'm wondering if there's some sort of like etymological
Yeah. So
back in the day.
I think the the behavior of goats jumping or doing silly things.
It's very interesting when goats will be walking along and suddenly they just start jumping, right? For no reason. Or maybe they have a reason.
They have their own reasons.
We don't know what
We're not privy to them.
They like to butt things too.
Yeah.
True.
And sometimes they break things.
And they eat stuff they shouldn't eat, like the corners of a fence or a tent or something.
Yes. Anyway, so they have lectins, which are carbohydrate-binding proteins that are from plants that are specific for alpha-2,3 or alpha-2,6 linked sialic acid, so they can stain the tissues and see what's present there. So they get a high expression of alpha-2,3 linked sialic acid compared to alpha-2,6 in the mammary cells, which is similar to what you see in bovine mammary glands. And they co-localize, they take some tissues from animals infected with mammary tissues infected with the turkey vulture virus, and they can see where the virus localizes with the alpha-2,3 linked sialic acids as detected with these lectins. Very little co-localization with alpha-2,6 linked sialic acid. So these viruses are entering cells using alpha-2,3, the avian type sialic acids, which is good. Yes. It means they haven't changed to use alpha-2,6 yet. They also made some air-liquid interface cultures from goat cells, respiratory cells of different types, from the nasal, the bronchial, and the small airways. So what they do is they have a cell culture dish with a membrane, and you have medium underneath, and then initially medium on top, and you plate out the cells, let them get confluent, and then you take the medium off and they differentiate into airway epithelial cells because the air is the key there for them to do that. So they can infect those and look at virus production, and they can see that the turkey virus is better at replicating, of course, all of these cell types compared to cow. And they also have a swine influenza virus that they use as a control. Same with nasal epithelial cells. But in general, the turkey vulture virus replicates to higher titers than the cow H5N1 virus. So all of these cells are susceptible to infection and give rise to infectious virus. The one thing that I thought here was interesting, let's see. The swine H1N1, which is not a cow virus, does not replicate well in these cell types. So that makes perfect sense. These viruses, also the cow and the turkey vulture, cause cytopathic effects. They kill the cells, and the H5N1 does not. And so they say that this is interesting because the swine H1N1 that they're using here, it prefers to bind to alpha-2,6, and there's plenty of alpha-2,6 in the mammary gland, but not replicating. So obviously other things has to happen other genetic changes have to occur in order to get replication in these goat tissues. They looked at antibodies in serum and mucosal secretions, and they find antibodies. They find more antibodies in animals infected with the turkey vulture, which they think maybe is because it replicates well, but they don't know what these antibodies do. They just they did some neutralizing antibody tests, but they're not sure what their relevance is to infection. And finally, they did some sequencing, whole genome sequencing of these viruses to see if they're changing in the course of infecting goats. They find some nucleotide changes over time, but none of them are concerning. None of them are what we think are changes that would lead to the ability to infect humans. Okay. So that's the story. So goats can be infected. They can be infected with cow H5N1 and turkey vulture. It reproduces a lot in the mammary glands. It gets into the milk, can spread to the offspring that way and infect them as well. And there's a little bit of a respiratory component, right? They have an interesting discussion here. They say acute respiratory infections typically start in the upper tract, right? You inhale the virus, it reproduces in the nasopharynx, and then it goes down to the lower in severe cases. But they had some goats here where they saw lower tract reproduction without any upper tract reproduction. And they're wondering if, in an industrial dairy farm, there's a lot of pressurized cleaning and stuff going on, and maybe this introduces virus deep into the lung of, say, a cow or even a goat. So they think respiratory transmission is possible, but probably is a less significant role than mammary gland.
But still be careful when pressure washing your goats.
Yes.
Yes.
And this, so this case, kind of one of the major inspirations for this study was this March 2024 case in Minnesota of a farm where the goats tested positive. And that they think for some reason that probably was from infected poultry. Yeah. So I'm guessing that would have been an aerosol transmission.
Yeah, they said probably there's some aerosol transmission.
Yeah, I mean, I think in any of these situations, if you have a farm with any of these animals, there's probably going to be some virus, high levels of that virus in the farm because of all of this.
Yes, H5N1 EIEIO. Yeah.
Then the final discussion is very interesting. They pointed that standard pasteurization will inactivate infectivity of H5N1 in milk. But then we have goat milk, which, as we mentioned, is an essential protein source in many Asian and European countries, especially India, Pakistan, Bangladesh, Turkey, and Italy. These are major producers of goat milk. Milk. These goats make a lot of virus, right? So I don't know when I was a kid, I went to my father's home in Italy, and they gave me goat's milk because they didn't have any cow's milk, right? But they boiled it on the stove first because they had their own goats and they harvested it. And I hated the boiled milk. I mean, I don't like milk anyway. And boiled, and it gets this film on the top. Yuck. But anyway, they're doing their own pasteurization. I guess.
that's a kitchen pasteurization.
Yeah. So they learned probably by getting sick from unpasteurized or unboiled milk. So the goats make a lot of virus in the milk, and so you have to be careful of that. Here in the U.S., I guess we don't drink that much goat milk, right?
Yeah, it seems like it's largely in those countries.
I've never seen it in the supermarket.
I very rarely hear about it. I did a quick search because I was wondering, as we were talking about this, what the relative number of goats versus cattle was in the world. If, the knowledge that suddenly there were goat infections meant that there were many more susceptible animals. And I learned that my guess about large numbers of goats or more goats than cattle was very mistaken.
We have more cattle than goats?
Way more cattle, yeah.
Okay.
How many cattle do we have in the U.S.?
I was actually looking at worldwide.
Yeah, that's the relevant comparison.
And let's see if I can get back to the number. So apparently there are about 1.6 billion goats or 1.6 billion cows and 1.1 billion goats.
Wow.
Okay.
So in the U.S., there are 94 million head of cattle and calves, with 28 million beef cows and 9.4 million milk cows. That's a lot of cows.
Yeah.
Yep.
94 million. Oh, I wonder how many pigs there are in the U.S. Let's look at that.
Let's see.
Number of pigs in the U.S. I bet there are even more. Oh, no, there are less. 75 million pigs.
Now, country by country, you may have, like in India, I could imagine you would have more goats than
right.
cattle.
That was sort of what I was thinking, especially since goats are smaller.
And in China, I would expect more pigs than
what do you
either.
think?
Right.
What do you think the number is in China? I'm looking at it here.
Of cattle?
Of pigs.
Of pigs. 50 million?
450 million.
Well, I had part of it right. Wow.
And let's look at the number of cows in China.
So they have almost 500 million pigs.
100 million cows, cattle.
Okay. That's a fraction of the pigs.
Any other animals we're interested in? How about goats? Let's see how they do with goats.
I don't think goats are that big.
The greatest of all time.
Yes.
139 million goats.
Okay, that's a lot of goats.
Right. Goats. Anyway, there you go. We have another paper for you. This one is in Nature, right? Yes, Nature. LRP8 is a receptor for tick-borne encephalitis virus.
Right, and this is from 4 co-first authors, Eva Mittler, Alexandra Tse, Pham Tu Hung Tran, Catalina Flores, and they only list one senior author, but there are 3 corresponding authors. So I'm going to say Andrew Herbert, Kartik Chandran, and Sarah Gredmark Russ. And this group is from Albert Einstein College of Medicine in New York, Karolinska Institute, Stockholm, USAMRIID in Fort Detrick, Geneva Foundation in Tacoma, Washington, University of Chicago, Institut Pasteur, Umeå University in Sweden, University of Iowa, Lund University in Sweden, and Linköping University in Sweden. International group.
So, well, full disclosure, Kartik emailed me and said, hey, you might want to do this paper on TWiV. Kartik has been on TWiV a couple of times, as has been Felix Ray, who's a structural virologist from France, and also Margaret Keelian. They have all been on TWiV. It's, as time goes on, we'll get more and more fractions of the authors.
Yes.
Yeah. Tick-borne encephalitis virus is a serious thing. Causes a severe neurological disease. Virus gets into the central nervous system, you get neuroinflammation. And as the name says, it is spread by ticks, in particular, ixodid ticks. Sometimes you can get it from drinking milk.
I was like,
yes.
Unpasteurized milk. It brings it all together.
Yes.
Yes, because the ticks bite the cow gets infected, and then the virus ends up in the milk. And infecting people.
Does the cow get mastitis? Is it a mammary
those
gland
are good
infection?
questions. Does this affect goats? Yeah.
It's a good question. But it's pretty rare, I understand, from what they say here.
Right.
Anyway, over 10,000 cases a year in Europe and northeastern Asia. So not really in the U.S., but the numbers are going up.
We have our own tick-borne infections here.
We have our own, yeah. And there are many infections, apparently, that are not clinically apparent. So there are lots of them. And there are vaccines, actually. There are tick-borne encephalitis virus vaccines in Europe and Russia. But, as you might expect, the coverage is pretty low. But if you live in those affected areas, it's a good thing, especially if you go outside a lot. If you just sit in front of your computer, you probably don't need to get vaccinated. But if you walk around, which Europeans are like, they like to walk around in the woods, I understand.
Yes.
You should get vaccinated. So anyway, TBEV is an orthoflavivirus. I have to tell you, when I went to Philadelphia for the textbook retreat, we took a yellow fever tour. So we had a docent, right, a young man who mostly talked about the early Philadelphia, because that's mostly what he knew, and he would sprinkle it with yellow fever stories.
Okay.
And he said, well, yellow fever virus is an orthoflavivirus. And we say, yeah,
good job.
Good job with that.
Nice. I learned a new word in this sentence, actually.
What's that?
Viscerotropic.
Yes.
That it was a viscerotropic disease. I don't feel like I've come across that one before.
So like liver would be a visceral organ.
Right.
Yeah.
I hadn't seen it phrased that way.
So other orthoflaviviruses dengue virus, Zika virus, of course, yellow fever virus, West Nile, Powassan. These are plus-strand RNA viruses with a membrane around the outside. There's a capsid protein encasing the RNA, and then around that is a membrane which has got glycoproteins in it, right? And the main one is the E-glycoprotein, which binds to the cell receptor, as we shall see. And of course, like every other virus, at least a mammalian virus, these viruses must bind to receptors in order to initiate infection. And so this paper is all about identifying the cell receptor or receptors for tick-borne encephalitis virus. And what they do is they have a cell line which is basically a CRISPR knockout library that individual genes have been knocked out by CRISPR-Cas9 treatment. And now you grow up a lot of these cells, and you basically have, in theory, different genes knocked out in every cell in the culture. And then you can infect them with your virus, and the cells that live presumably have deletions of genes that are essential for virus multiplication, not just receptors, but everything else, because as you know, viruses need many, many genes and gene products from a host cell in order to reproduce.
You probably have to infect with a really high MOI.
I would think.
Yes. Yeah. And this is a way to test the whole genome, essentially. I mean, there are some genes, you knock them out, the cell's not going to survive. But right. You can search through the whole genome at once to find out what the virus needs.
And then you have these surviving cells, and you can identify which gene has been knocked out, because the CRISPR-Cas9 introduces kind of a barcode, and as you sequence, you can amplify using the barcode, and then you can say what gene, because the CRISPR-Cas9 doesn't take out the whole gene; it just disrupts it, right, to make it non-functional. So they do this, and they have a whole bunch of cell genes that, when disrupted, make cells resistant to tick-borne encephalitis virus, and they put them in groups. And they have different functions, like some genes are involving targeting viral transcripts to the ER. Some genes are associated with the ER-associated degradation pathway. So the implication is that would restrict virus reproduction. So you disrupt it, the virus can replicate. Lipophagy, RNA binding and replication, and the synthesis of cell surface glycans and proteoglycans, okay? So they find a lot of different categories. But they find a single hit that is strongly enriched, which encodes a protein called LRP8. And it encodes low-density lipoprotein receptor-related protein 8.
And this is they found other hits that were as good that were in other, like the ER membrane complex, but LRP8 is their best hit of cell surface proteins.
Right.
Since they're hunting a receptor here.
Yeah. So it's this protein has been recently identified as an alpha virus entry receptor. Can anyone name an alpha virus?
Chikungunya,
Semliki forest, Sinbis, yeah. Did you hear that there may be a local chick case on Long Island?
No.
So a lady was presumptively diagnosed with chick. They have to confirm it, and she never went anywhere. Right, because you would typically go somewhere and acquire it, then bring it back. So maybe there's chick endemic on Long Island.
Oh, dear.
We'll see.
Or at least a transmission event.
They have to confirm it.
Yeah.
Maybe they have already. Let's see if they've confirmed it.
Chikungunya,
Long Island. No, not Chick-fil-A. No. You have to write out the whole word. Chikungunya. A possible locally acquired case. 60-year-old woman in Hempstead tested positive for the virus in August. And she had severe joint pain, which is a symptom of the disease, and did not have any recent history of international travel.
So now we have locally in this general area, local chick and local malaria.
Well, now
I'm going to have to not go outside.
I'm not finding stories that it's been confirmed yet.
No, it doesn't seem to be confirmed yet.
Still possible case.
Yeah. Anyway, those are alpha viruses, yes. And so they said, all right, let's see if this is a receptor for tick-borne encephalitis virus. So they take a normal cell. So they're using A549 cells, human lung cancer cell line. And they use CRISPR-Cas9 to knock out specifically just LRP8 gene. Okay, nothing else, right? And they have two different knockout lines. And they say both have reduced susceptibility to entry. Reduced is not zero, but it's really low. If you look at the figure, it's pretty good, but there's still a little bit of...
And it's statistically significant.
Mm-hmm.
Yeah, that's what I'd need to convince me, right? I'm just kidding. I don't give a damn about statistics. What is if I need statistics to say my experiment means something, I don't want to do that experiment. But that's because I was trained to do laboratory cells in culture infected with viruses, and you have 10 logs of poliovirus and no logs of polio. I don't need no stinking...
Sometimes you need the statistics.
There are some situations where statistics are important.
Yeah.
Yes, but you don't need them to convince you of a biological relevance.
If something is very biologically relevant, you do not need it.
It will be clear, yes.
I understand in like population studies and so forth like that, but don't you agree, Alan, the kind of experiments we did?
No, I never used statistics in the lab. I mean, if it wasn't a log difference, it didn't register.
Yeah, we didn't want to work on it. Anyway, so this knocking out LRP8 reduces susceptibility. And they're not actually using the actual virus. They're using reporter virus particles, right? They have they're viruses that have the glycoprotein but they don't actually do a whole replication cycle and make infectious virus. They just get in the cell, which is a receptor-dependent step, right?
Yes.
And they also look at other TBEV subtypes. So they're working with one main one here, the Neudorfal TBEV, but they also look at the European Far Eastern subtypes, and they are also reduced. So it looks like one receptor rules them all.
Yes. Also the Langot virus chimera.
Yes. So that's from the highly neurovirulent European TBEV strain. And then if they put LRP8 back into those cells, then they will now be susceptible to TBE, which is an important experiment because you don't know if your CRISPR did some off-target chopping, right, which is actually what's going on. But no, they confirmed that it's LRP8. These A549 cells have pretty low levels of a LRP8, and when they overproduce LRP8 in those cells, you get a good increase in entry of TBEV.
And that change you don't need statistics to see. Pretty clear.
No. You just have to be careful. What is it called? The tyranny of the P factor, right?
Yes, right.
The P value.
Mm-hmm.
If you have to keep massaging the data around until you get a desirable statistical significance, then you're doing it wrong.
Yeah, some people will think that just because the p-value is good that your data means something. No. It just means that it's statistically significant. It doesn't mean that it's biologically relevant, right? Okay, what else do we have here? So they looked at E proteins of a variety of orthoflaviviruses, what they call authentic orthoflaviviruses. I think that's funny, right? As opposed to the Confederate ones, the counterfeit ones.
The counterfeit ones.
And interestingly, only TBEV seems to be binding to LRP8 among the authentic orthoflaviviruses, but overexpression did boost infection by Semliki forest but not Synbis. But these are alpha viruses, so that makes perfect sense. So it seems to be unique to TBEV to bind LRP8, right? So then they asked, well, is it just LRP8? There are other homologs of LRP8. In fact, there are 13 homologs in cells, so related proteins. And so they knocked each of those out and made cells and infect them with their tick-borne encephalitis bearing what they call them reporter virus particles, and only LRP8 works to allow virus entry. All right, what part of the virus is binding? This is a classic virus receptor identification paper. Now, what part of the virus? Well, we know that what part of the LRP8,
right,
is required. It has different domains which have been characterized, for example, an N-terminal ligand binding domains, because this receptor has a normal ligand in the cell, and it has 7 repeats. It has 2 epidermal growth factor-like domains. It has a 6-bladed beta propeller. I love that. That's why I said it, the 6-bladed propeller.
Beta propeller.
Yes, which they also show very nicely in their little figure.
Yes.
A 30 EGF domain, a membrane proximal serine threonine rich domain, and a transmembrane and cytoplasmic domain. Right? Yeah, they put a little propeller there. And so they actually, their approach is to overproduce each of these and see if it makes a difference if it interferes with infection, right? And basically these repeats, these repeat domains that I just told you about. That's up further. I went too far down. The LDR Class A repeats. 2 of them, 1 and 2, are probably, they conclude, are probably directly involved in entry of TBEV. So then they take the domains, those 2 parts, 1 and 2, a small part of the whole receptor, and they make a soluble form of that protein, and they try and see if that binds virus particles. And they did. They do 2 assays. They do a bead capture assay and an ELISA assay, and they can show that both 1 and 2 will bind to the virus. And it's specific for the glycoprotein E of TBEV. Okay. And they don't see any binding to Japanese encephalitis virus.
And this seems really interesting because this already starts to suggest what would happen if you made this as like a pharmaceutical product or something like that to potentially help with this virus.
So in these experiments, they have a monoclonal antibody against TBEV E protein, which can block the interaction with the cell receptor. And this monoclonal is binding to a different part of, is binding to domain 3 of the E protein. So the E protein also has multiple domains, and this antibody is binding to domain 3. So they're wondering if that's involved in binding to the receptor, right? So what do? They make domain 3 of E only, but it doesn't bind the domain 1, 2 of the receptor.
Right.
Doesn't bind, even though it should have. And so they think maybe other parts of E may be involved in the ability of this part to bind. So they make a, they basically fuse it to a scaffold, which is a pentameric disulfide linked scaffold derived from an entirely different protein. So not TBEE at all, from a cartilage protein. And then they can show that the domain 3 of the E protein will interact with these 2 repeats of the TBE, right? So basically you need to have domain 3 in some kind of higher order structural configuration. So they conclude that LRP8 and E interact with multiple contacts between these 1, 2 domains, and the rest of the rest of the LRP8 and also the domain 3 of TB8 have a key role. So what part? So we see now that LRP8 is needed for entry, right? But entry comprises binding and entry into the cell, right? The virus particles bind a receptor and then they're taken up into the cell. So where is this protein involved, because it could be involved in entry. We haven't done any binding assays yet.
Okay.
Right? So let's do some binding assay. They incubate cells and virus at 4 degrees Celsius, and then they stain for the E protein. So 4 degrees, the virus will bind and it will stay on the plasma membrane. It will not enter because at 4 degrees, you have shut down endocytosis. It's a 30, 40-year-old experiment, even more. I remember when I was a graduate student learning this, and you could warm up the cells and boom, it would go inside of it. Maybe Alan even did
I
that.
did a lot of that in my viral entry study. Yeah.
Yes. So they find and cells that don't have LRP8, there's no fluorescence of the virus on the E signal on the cell surface. But if you do this experiment where you have produced LRP8 and now you see lots of cell surface E staining, so you can say that LRP8 mediates attachment to cells.
Yes.
By that experiment. Yeah. And then they do the same experiment and they then incubate them at 37 degrees for 2 minutes to allow the virus to get into cells. And they can see, they do the time course of this, and they can see that the E protein goes from the plasma membrane inside of the cell, it goes into endosomes and the endocytic pathway. And so you can see that's dependent on LRP8 as well. So you can see that the binding is what is happening first, and then that binding takes up the virus particle into the cell. And if you look at susceptibility of different cell lines, you see that there's a concordance between susceptibility and the ability to support this attachment and internalization that we've just described. Okay, now getting back to something Brianne said, can we make a soluble receptor which might be a decoy and block infection? And so they make soluble domain 1,2 of the receptor linked to an FC domain of an antibody, and that will reduce virus entry in a dose-dependent manner. So it's basically neutralizing infection. And you could think about making a therapeutic, but people have tried that, and it's not great with other viruses. TBE, as you know, we said can infect the brain. It can infect neurons and glial cells. So they make some neuronal cells, like basically neurons, from human pluripotent-induced stem cells, and they show that they have LRP8 on the surface, and they can be infected with the virus, and that their soluble domain 1,2 of the receptor can block that infection. So it's the same receptor used in the brain. And finally, a mouse model. They have a mouse model for TBEV pathogenesis. Is LRP8 involved? Is the question, right? There's an ortholog in mice, a mouse LRP8, very similar to human LRP8, 87% amino acid sequence identity. Although that's a lot, that could be enough. That's
a pretty big difference.
Not work it, but it works because they can produce that mouse protein in A549 cells, and it boosts TBEV infection. So the mouse ortholog works. So they have a model where they infect mice and they rapidly develop clinical signs, and they essentially die. They have neurological disease. And so they can give these mice first some of the receptor decoy and then challenge them, and they survive. 19 out of 20 mice survive. They don't develop clinical symptoms.
Which is pretty good.
It's really good.
Although the reason those types of approaches have failed in the past are typically twofold. Either the decoy ends up having some other biological effect that you don't like, or, they can both happen. You put this in and the virus quickly produces escape mutants that can somehow get around your decoy.
Brianne, hasn't recently a CD4 decoy been used for HIV?
It has been used some. There are some CD4 independent viruses though, so it's not gonna work for everything.
I think it's part of one of the approved therapies. Initially, it didn't work years ago.
Right.
and now they've gotten it to work. I'm not sure why. I haven't looked into it. Anyway, with this model, so the decoy reduces viremia in the mice, and it reduces virus infection in the brain. And you can give them this 6 hours before infection. Now, that's not so useful to.
You don't have 6 hours advance notice before you.
Typically,
get
you have people with symptoms already.
Yeah.
Which may be too late to give them a decoy. But anyway, that's another receptor identification, and they say, well, maybe there's some other entry factors because if we knock out LRP8, you still get a little infection, so a little more work to do.
Yeah. Right? I was looking at the Human Protein Atlas to see where LRP8 is expressed.
Yeah.
Because as I was thinking about this, my guess was, and I think Vincent would probably agree with me on this, that it is unlikely that this virus evolved to use LRP8 because it so desperately wanted to go to the brain.
Right. It's in it, yes, for sure.
And so, in theory, LRP8 has to be somewhere else that the virus would find useful. As I was trying to figure out where it was, I could sort of tell, at least from their A549 data, that lung perhaps was not the answer. So it's interesting to look at. I don't have a clear answer, but.
Well, I would say that it has a viremic phase, right?
And in fact,
the ticks take a blood meal and they pick it up and transmit.
Yeah, and that was my thought, was that that the lymphatic system and different immune cells were not the highest, but were definitely measurable here, and were probably the answer.
And I'm going to hazard a guess that maybe it's in the liver and some other visceral organs.
Yeah. Anything that will give you a viremia.
For the viscerotropic.
Yeah, actually not so much liver as you would expect. Thyroid and testes are the big ones outside of the brain. And in general, various endocrine tissues.
Yeah, I think in general, when viruses get in the brain, it's an accident.
Oh, yeah.
Right.
Because the receptor happens to be there, right? So poliovirus receptor is mainly in the gut, and that facilitates transmission by fecal contamination, but it's also in the brain.
Mm-hmm.
So it gets in the brain because a virus in humans is, once it gets in the brain, that's it. It's not going to transmit to another person. And, transmission drives evolution of viruses. So it's an accident.
Yeah. So it is relatively highly expressed in the brain. The brain is, not the highest, but it's pretty high here. But I think I was guessing the immune cells and the blood meal, like Vincent said, are probably the reason.
Although a virus that wants to infect zombies would probably do well by infecting the brain.
Because they eat each other?
They eat brains, right? Isn't that part of the trope?
Well, animals, non-human animals eat other animals, right? So you could imagine transmission by that. But
I'm not thinking viruses at anybody.
The last brain consumption I heard of in humans was the 4 people of New Guinea, right? And how they transmitted Creutzfeldt-Jakob disease. Kuru, sorry, that's the name of the disease, Kuru. Okay, let's do some email, and I think it's appropriate that Alan take the first one.
Okay, which was the first?
It's written by Alan.
Oh, yes. Alan writes, this is a different Alan. On Tuesday, Pete Hegseth addressed the general officers and senior enlisted personnel of the entire U.S. Armed Services. In my mind, his most concerning comment was about his plan to remove consequences for refusal to accept, quote, an experimental vaccine, end quote. If it is so critical to adhere to grooming standards and avoid beards, unless protected by earning a Special Forces shoulder tab, it is nuts to allow soldiers to refuse vaccinations that are shown to be safe and effective. Point in fact, the leading cause of death for the U.S. Army Expeditionary Forces in 1917 to 18 was influenza and its related complications, gives a reference. A refusal of vaccination by 10% of a tightly packed group can overwhelm the protective effect of vaccination, since about 3 to 4% of any human population randomly fails to produce a protective immune response after vaccination. And Alan is an MD. I would add, actually, that this line about removing consequences for accepting a quote-unquote experimental vaccine is a bit of stage trickery going on here, because in fact what happened when the COVID-19 vaccines were rolled out under an experimental authorization, which was done because there were developed in 9 months and there wasn't time to get the full authorization paperwork done. They were offered to the armed forces, and my understanding at the time was that they were actually voluntary, at least initially. And so the troops were not mandated to take this experimental, if you want to call it that, vaccine initially. Now, whether that changed, I don't know, but I think this is just a bit of, throwing more red meat to the base.
So this doesn't affect the approved vaccines that recruits get, right?
No.
Right. I think it's just the EUAs.
It's worth pointing out that recruits get some vaccines that the general population does not get, right?
Smallpox.
So they get smallpox, and they also get an adenovirus vaccine because when you throw recruits together in the beginning, they tend to get adenovirus outbreaks, which we don't see a kind of outbreak in humans.
Which decreases military readiness if you care about that.
Mm-hmm.
Yeah. If you're lying in bed, you can't
be out fighting.
You're supposed to do. Brianne, can you take the next one?
Sure. Jay writes, "Hi all, Jay here again, responding to Rich's questions about my hemophilia gene therapy treatment that I brought up in my letter you read out on episode 1257. Yes, the treatment used an AAV capsid vector with a gene cassette containing a factor IX gene and all that cool virology stuff that optimizes it to work in hepatocytes where factor IX is naturally produced. As I understand it, the AAV capsid vector is long gone after a few months, and the deposited genes are just floating around episomally, producing factor IX. Thank you very much. I was on a clinical trial run by the amazing people at CHOP in 2016, so no, guinea pigs don't have to pay. And being on a trial, yes, they took a lot of blood for the first several months, then less often over the next 6 years of follow-up. The factor IX expression has been very stable. Now, no one is following up, but 9.5 years on, the proof is still in the pudding and I don't dwell on it much. That's the whole point, isn't it? Fun fact, the factor IX gene that was used for this gene therapy was from a specific family in Padua, Italy, that have a hyperactive factor IX gene mutation, causing thrombophilia. So the gene therapy dosage could be relatively low, 5 × 10⁻11 viral genomes per kilogram, minimizing the vector immune response risk. Not so fun fact, this therapy was improved for use in Canada in 2024 and discontinued in 2025, apparently due to limited interest. The price in Canadian dollars was set to be $4.7 million per patient. If I may Racaniello for a moment, I can't but think that curing patients is not a sustainable business model and not as desirable as treating lifelong chronic diseases. $300,000 to $500,000 US per year for a hemophilia patient, for example. I think especially now with curative treatments becoming a reality, licensing publicly funded intellectual property such that it comes with usage obligations, use it or lose it, needs to be regulated somehow. Way above my pay grade, I'm just a virally infected gain-of-function mutant, not something I'll brag about at the US border next time I visit. Thanks again for all you do. My pick of the week is the 2006 film Who Killed the Electric Car? Jay. And Jay is from Cambridge, Ontario.
I would argue that a health insurance company would rather pay $4 million than $500,000 a year for your lifetime for treatment, right?
Maybe we need an actuary to weigh in, or an accountant at least to weigh in on that. Because if you're paying out a few $100,000 a year, you keep the rest of the $4 million in your float earning interest.
I see.
I'm not sure you'd come out ahead that way, but the company might care.
I don't think this is right. I think people's health is what matters the most.
Yeah, I think so too.
Not the profits of companies.
Especially when, unfortunately, there's also the third option of you'd also do pretty well if you denied both.
Yes.
And I definitely don't think that
financially
we want anyone to do that.
Yeah, the insurance companies would be all okay with that.
Daniel and I talk about this a lot because a lot of hospitals will not dispense Paxlovid. And Daniel says, well, that's because they have to pay for it up front, and then they're not going to get reimbursed very much. So they're really making a clinical decision based on the insurance company, which is unfortunate. Right?
That happens a lot in American medicine.
I'm sure. Daniel always says that it's better to take Paxlovid instead of and pay for that rather than going into the ICU for a week, which is a higher cost than Paxlovid.
Yeah.
Mm-hmm.
But the economics of life, oh my gosh.
Well, the economics of U.S. healthcare anyway.
Yeah.
So it's better in other countries?
Well, I mean, the Canadian situation is probably a product of the company that was doing this. I mean, Canada regulates their drug prices, but maybe not for a situation like this where there's a very small patient population. The pharmaceutical development's happening in the U.S. To a large extent, and the companies are charging what they like here.
Yeah. All right. Robert writes, I'm writing regarding your comments in episode 1257 about the unfortunate U.S. government announcement about acetaminophen and autism. You stated a few times that there's no association between acetaminophen use during pregnancy and autism. This isn't correct. Yes, you're right. There is an association. There's no causation proved.
As I've looked at this letter before this, I am teaching epidemiology for my first time ever this semester. And I was thinking, thank you so much, Robert, for putting this together for me to use in my class later this semester.
Yes. Copy, paste, yes.
All right, so you're right. This isn't correct. There seems to be a fairly robust association from the Ahlqvist paper, the 2.5 million children Swedish study. Quote, crude absolute risks at 10 years of age for those not exposed versus those exposed were 1.33% versus 1.53% for autism, which translated to a statistically significant hazard ratio of 1.05. I remember hearing an interview with one of the authors of this papers where he mentioned that he was relieved to see this initial result because it meant their study sample was consistent with previous reputable studies on the subject. We use the word association for a reason. Association means that two variables are correlated. Contrary to internet contrarians, correlation does not imply, correlation does, well, he says
does
right.
imply a causal relationship, but it does not specify the type of causal relationship.
Okay. There are three possibilities. One, acetaminophen use causes or contributes to a baby having autism. Two, the fetus having autism causes mom to use acetaminophen. Or three, some other factor causes both. We say we observed an association because we don't know which of these possibilities is the true relationship, and we shouldn't jump to the conclusion that it's the first one. Earlier papers were clear about this. Simple correlations are relatively easy and cheap to observe, so they're a good starting point. They indicate that there might be an interesting relationship, and it's worth spending more time and money to figure out what it is. Ahlqvist that I'll take the next step. After confirming the previously observed simple association, they used their enormous sample size to compare pregnancies with and without acetaminophen exposure within families, comparing siblings, one of whom was exposed to acetaminophen and the other wasn't. And in this comparison, the association between acetaminophen and autism disappeared. Acetaminophen use during pregnancy was not associated with autism within families or equivalently between siblings. This result suggests the causal relationship is type 3. Differences between parents cause both acetaminophen use and an increased chance of having an autistic child. Ahlqvist et al. Couldn't say what this parental characteristic is. That will require further research. But autism has a strong genetic component. Autistic parents are more likely to have autistic children, and autistic people are also more likely to suffer from other conditions, such as migraine, for which they are likely to use acetaminophen. More concerning is that autistic mothers may be more likely to get fevers. Untreated fever during pregnancy is known to increase the risk of neurodevelopmental problems in the fetus, and acetaminophen is one of the few treatments for fever available to pregnant mothers. Avoiding acetaminophen could increase the risk of neurodevelopmental disorders, possibly including autism itself. Association isn't a sciencey word for causes. The term is used specifically to indicate that we can't yet say what the cause is for an observed relationship. I think it's important to make this clear in scientific communication to the public, but also important for scientists to periodically remind ourselves that many of the words we use are used for specific and important reasons. Thank you for all you do. TWiV and its sister programs have kept me entertained and informed during tens of thousands of kilometers of travel over the last 15 years by car, plane, and boat. Robert. Oh, dear. That's lovely.
Excellent.
That is, like I said, so wonderful, Robert. And thank you so much for the fact that I will be stealing some of this exactly for my class. Yes.
Yeah, you should use it. It's perfect.
We have talked already about sort of some of the details of association. And I use the example of there is a really nice association between how many people drown in a month and how much ice cream is eaten in the month. And so you might say, well, does ice cream cause drowning? Does drowning cause people to eat ice cream? Or is there another third factor?
Right. And yet that makes you do another study, a big study like the Swedish study, which not everyone can do.
Exactly.
Right.
There's a strong association between carrying matches and developing lung cancer as well.
Yes.
Let's do another round, Alan.
Okay. Ted writes, I'm back. I was a faithful viewer during the COVID pandemic in 2021 to 23, and then with vaccines, I stopped weekly watching. With this absurd environment promoting questionable science, I'm back to weekly watching. You are a beacon of light and truth. Please stay strong. We need you. I got my COVID and flu vaccines here in South Carolina. I am 80 and had to get a prescription from the MD for Walgreens to give us the vaccine. Another hoop to go through. P.S. I still donate even when not watching. My insurance payment. Ted is in South Carolina. Thank you, nice. Ted.
Welcome back.
Yes.
Brienne.
Sure. Claire writes, greetings. I'm a fan of your show to try to stay on top of what is what vaccine-wise, especially in the current climate. It is becoming increasingly difficult to contextualize information properly. So thanks again for putting it out there. In episode 1254 at minute 29, you talk about the risks of complications with COVID-19 infection in pregnant versus non-pregnant women. 3-fold risk of acute myocardial infarction, 5-fold risk of pericarditis. I looked up the article as I wanted to use some stats to promote vaccination uptake in our pregnant population here in Nunavik, northern Quebec, homeland of the Inuit. I'm by no means an expert in adjusted odds ratios, but I believe the risks are actually similar between pregnant and non-pregnant women when they catch COVID, and the adjusted odds ratios instead compare incidence of events in pregnant infected versus pregnant non-infected women. Quote from the article, more adverse events occurred in COVID-19 diagnosis, including pericarditis, pregnant adjusted odds ratio 5.35, 95% confidence interval 2.24 to 12.79, non-pregnant adjusted odds ratio 5.57, 95% confidence interval 3.02 to 10.27, and acute myocardial infarction, pregnant adjusted odds ratio 3.15, 95% confidence interval 1.56 to 6.37, and non-pregnant adjusted odds ratio 4.34, 95% confidence interval 2.79 to 6.76. Keep up the great work, and best regards, Claire Elise, who is from Quebec. And yeah, I believe you're right, Claire. I have not looked at this article, so I cannot say for certain, but I believe that the odds ratio is about the incidence of these adverse events.
Yes, so it doesn't make sense to not allow pregnant women to get COVID vaccines here in the U.S.
Right.
Or anywhere.
But most countries recognize pregnancy as a risk factor, right? John writes, enormous thanks for so much great information every week. It's great to be reminded that there are still serious people doing serious science. A heads up, you've noted the many cases of measles in Canada, but Mexico also has a big outbreak clustered hard by the U.S. border. As of Tuesday, Mexico's government had confirmed 4,653 cases and 21 deaths, with a total of about 10,000 cases listed as suspected. Most are in Chihuahua state bordering Texas. A link to the data on a government website with a map, and he provides a link to that. Many thanks for everything. John is in Mexico City. I didn't realize that there was a big outbreak there.
I did not, and that's terrible. I saw that there was another death today in South Carolina.
Really? Really? What age?
I just saw child. I think it was pretty young, but I don't remember exactly what it was.
And that's interesting that it's, interesting, not surprising that it's clustered right along the border with Mexico and the U.S. So apologies to our Mexican colleagues that you have to live next to this banana republic that's run by an anti-vaccine dictatorship at the moment.
Wow, I'm glad to hear your strong words, Alan. You're next.
All right, David writes, "Hello, I'm a passionate listener and learner from TWiV since May 2021. Here is an informative article today about measles and how it spread so fast and far in Canada and the US. As a guy almost 78 years old, I cannot understand why younger people disregard science and the hard lessons learned from years ago, as well as the miracle vaccinations and drugs developed and used widely over the years," and provides a link to this article, The Globe and Mail in Canada. And it's talking about the... actually, I'm not loading the article.
Yeah, I can't get it. It seems to be paywalled.
Yeah.
It's called How a Single Case Became 5,000 or something.
Yeah.
Like that. But it's not just young people, though, right?
No.
I mean, RFK Jr. is not young.
No.
Neither is the president.
No, definitely not.
But I do agree that people who have never seen infectious disease outbreaks are sometimes not impressed by the need to vaccinate.
Yeah.
Brianne.
Lori writes, "Hi, friends. I have listened to all the TWiV episodes, but decided that I would go back and listen to some of them again. I thought of this after enjoying your reposting of episode 100. Today, while doing some house cleaning, I listened to episode 504, Flying Foxes and Barking Pigs. It was so nice to hear Dixon's voice again. In the episode, you discussed Nipah outbreak in India and the first case of polio in Papua New Guinea in 18 years. During the discussion, Vincent mentioned that micropatches were being tested for IPV injection replacement. I am wondering about the result of those tests. I assume that the patch did not give similar results to the injection. Is that correct? Another point that was made was that to have enough IPV for the entire world, there would need to be huge batches of live virulent virus grown for manufacturing, and this would also be a major problem. Would using mRNA for a polio vaccine help with this issue? Guess research couldn't be done here for multiple reasons. I am really enjoying the older episodes and recommend it for other TWiV fans. Keep on TWiving, Lori."
Oh, yes. So, yes, the micropatches were tested. They don't give as good as an immune response as IPV. So I don't think they're going to go anywhere, unfortunately. And so, yes, growing a ton of IPV is somewhat problematic. We did a paper where they, if you remember that in Belgium, a vaccine facility accidentally released virus into the water, right? So these things can happen. Accidents can happen. So it would be nice to have a non-infectious...
Nobody was hurt by that, by the way.
Nobody was hurt by it, yeah. So it would be nice to have something else, but you can't do research on polio any longer. So we can't make new vaccines. Well, you could if you just did subunits, but it would be hard to test them, right?
But also yes, mRNA could potentially be really interesting, but also can't really do that.
Yeah.
Right. All right. One more is from Nick. Thanks, Vince, Allen, and Rich for rebroadcasting this superb interview. That's episode 100 with David Baltimore. Seems as if it could have been done yesterday. It was so relevant, and he expresses himself so simply and clearly. I don't know if you know, Alan, I re-released that.
Yes, I saw. It was a great idea.
And I had found some video that I had taken and put that up too. Why not? He had just left the TAM Chopin Lab in Founders Hall of Rockefeller before I arrived in fall of 1963 to start my PhD. He finished his PhD in 1964, but it took me some time to finish mine. I began working on a poliovirus project suggested by my mentor, Igor Tam, but left it to a postdoc in the lab while I was taking some time to learn German and do some traveling in Europe in the summer of '64. David was so smart and ambitious and knew where to look for hidden pearls in molecular biology and virology, one of the best. And that's Nick Atchison, who has often written. He used to be a virologist up at McGill, if I remember properly. Thank you for writing. I really enjoyed, his answers were great.
Yeah.
It was such a good episode.
Yeah, that was a great episode.
Mm-hmm.
It was back when we used Skype.
Yeah. That was our first big milestone episode, 100 episodes. So, and having David on was a big treat.
Time for some picks of the week. Brianne, what do you have for us?
I have an article that I read talking about astronomers potentially finding some clues that have suggested to them something called Planet Y. And I have read in the past some discussions about previous suggestions about Planet X. Basically, we can't see it, but it seems like in some situations there is something gravitationally pulling on Neptune and some of the outer planets in the solar system, making us think that there might be another planet that we have not yet been able to see. That is actually how both Uranus and Neptune were discovered, is that first someone said, hey, orbits aren't really right of things. There must be something else pulling. And then they looked and found another planet. So people have been talking about Planet X as this additional planet that might be pulling on the orbit of Neptune for a while. And now some additional data has come out suggesting there might be yet another planet influencing orbits of Kuiper Belt objects and things in the far solar system that they have named Planet Y. And I find all of this really fascinating to think about how we don't necessarily even know how many planets are in our solar system, and how many sort of observations and things there really still are for scientists to learn. Yeah. And how we can figure out those things about things we can't see. Here, of course, it's the other direction, not the viruses that we can't see because they're too small, but things that are too big and far away, and we still have to use really interesting science to find them.
We don't know all the immune cells. We don't know all the planets.
I know.
Yep.
Oh my gosh.
So much to learn.
But what they did to Pluto was unconscionable.
It really was.
Yeah.
But I guess they know what they're doing, right, Alan?
Yeah. I mean, the astronomers do have a point. It's a Kuiper Belt object that there are larger ones out there. So if it's a planet, then is some other rock out there a planet?
Yeah, and then do we have 1,000 planets? How are the second graders going to cope with that memorization?
Could you imagine if they said this virus is no longer a virus? I don't know what else it would be, but that would be bad. I would object. I would lobby against that.
I mean, it's maybe not as bad as what they did to Uranus, but it's it's a pretty raw deal for Pluto.
Alan, what do you have for us?
So October has started, and my neighbors are all putting up their Halloween decorations, and so I'm pulling out a pic that I actually have saved for a few months just for this occasion. This is I'm linking to the Science news story about it. There's also a paper linked in, but really all you need to do is watch the video. So this is a video showing a microscope. It's like a dissecting microscope magnification level of a Caenorhabditis worms, soil nematodes that have formed a superstructure. So it's like a finger made of worms. It's this thing reaching out, and it acts and moves like a single organism. So it's a tower of worms moving in coordinated fashion, and these worms are able to move themselves around. And you see in the video, you see a couple of stimuli of this thing, and it responds to it. So that's a little something to watch, and
watch till the end.
Yes.
See what happens to the Drosophila.
Yes. Yes, it grabs the fly. So I thought that was fun.
Neat.
And there's cool science to it too, because these worms are coordinating their actions almost like they're a multicellular animal, but they are themselves multicellular animals. So they're this meta-organism.
Neat. My pick is an article in Wired, which I've just learned now I've expired all my Wired free article reading, so I can't look at this, but it's called "The EPA is Ending Greenhouse Gas Data Collection. Who will step up to fill the gap?" So the EPA used to require polluting companies to track their emissions, and that would help form climate policy. But now the EPA, of course, is being dismantled, and maybe it's going to be eliminated. Who knows? And so it doesn't require these companies to submit emissions data anymore. So this article is about whether other non-governmental organizations could step up to do it, whether they're technically able, but also legally. Who knows if they're legally able to require companies to do that? They may not be. So if you're concerned about these sorts of things, and you should be, you should read this if you can.
Yeah.
But it is paywalled. We have 2 listener picks. We have one from Jay, who we heard talk about his hemophilia correction. And his pick is Who Killed the Electric Car, which is a movie, but I didn't know it. And I put here the Wikipedia entry. It's a 2006 documentary film that explores the creation, limited commercialization, and destruction of the battery electric vehicle in the U.S. in the mid-1990s. Of course, since then, we have embraced battery-driven vehicles, but at the time, they were killed.
Mm-hmm.
And so this explores, if you're interested, this Wikipedia article explores that. So GM wanted to demonstrate that there was no consumer demand, right?
Yep. I saw this this documentary back in 2006.
Yeah. So it was a fabricated story, right? They just wanted to block competitor cars.
Yep.
Right.
And also Douglas. Hello, TWiV. This parody song by the Marsh Family may bring a little cheer. Measles and polio down in the schoolyard. This is really good, folks. First of all, they're very good. There's a whole family singing and playing various string instruments. And the lyrics are great. And, the original song is just awesome, right? The Simon and Garfunkel song, or Paul Simon. But this is really good. So at least one musical family recognizes the folly of RFK Jr. It's good to see that.
Yeah.
All right, that is TWiV 1259. We hope you enjoyed it. If you did, would you like to help make more of these? We'd love your support. All you have to do is go to microbe.tv/contribute. There are multiple ways you can do that. It doesn't cost you a lot of money, and it really gives us a lot of support so that we can make more of these programs. You can find the show notes for this episode, all the links that we've mentioned, at microbe.tv/TWiV. And if you have questions or comments, if you have a pick of the week, we love reading your email and your questions and your comments and your picks. Please send them to twiv@microbe.tv. Alan Dove is at alandove.com and turbidplaque.com. Thank you, Alan.
Thanks. It's always a pleasure.
Brianne Barker is at Drew University, Bioprof Barker on Blue Sky. Thank you, Brianne.
Thanks. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv, and Microbe TV is also on Blue Sky, Microbe TV without a dot. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene Ramsey for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.